So welcome to the Sparks Nature Center, everyone. My name is Theo Lagarde. I serve as the Gunpowder River Keeper, and I'm pleased to have Aiman Raza here to discuss her work towards her master's thesis at UMBC Eye Care, and also want to acknowledge that her professor, Tamara Mendelson's here. And we're so happy to have the cohort here, her peers and family and friends of the Gunpowder River. Sparks Nature Center dates back to 1915, but it was not always a nature center. We have 1 million people along the trail each year that benefit by this water being a protected state and the fact that it's drinking water for 1.5 million people. And I believe Aiman's research is going to really heighten awareness not only of brook trout, but how sensitive they are and how valued they are to residents in the upper watershed. Thank you.
Mm-hmm. All right, hi everyone. My name is Aiman Raza. Like Theo mentioned, I'm a biology master's student at the University of Maryland, Baltimore County, and also part of the eye care program. So today I'm going to be giving a talk about forest to faucet safeguarding clean drinking water through brook trout science and conservation. So before I go on, I did want to start with a land acknowledgement. So this is the Gunpowder River Keeper Land Acknowledgement Statement. So the Gunpowder River Keeper acknowledges that we protect land, water, sensitive species, and communities in the traditional territory of many nations, including the Algonquin, the Susquehannock, and the Delaware peoples. So as you guys were coming in, you might have seen this QR code going around. So this leads to a little quiz that I developed, testing your knowledge about brook trout. It's titled, "Are You Smarter Than a Brook Trout?" So, everyone gave it their best shot and answered the questions on there. And hopefully by the end of this presentation, you guys will be a lot more well equipped to answer these questions. And we will be retested at the very end. So pay attention. All right, so I want to start off by asking everyone, how often do we think about the water that's coming out of our faucets? We have the privilege of having access to clean, safe drinking water. But we rarely think about the journey that water takes to actually get to us. And we're not the only ones that rely on clean water. Also, the many fish species that live in our local waterways also depend on clean water. So water connects all living things, and through this presentation, we're going to explore that connection, and we're going to find out how we're actually connected to brook trout more than we might expect. So this is a little outline of what we're going to be talking about today. So the first part is brook trout biology and habitat. So basic facts about brook trout, where they live, where they're found, some issues that they're facing. And the second part, we're going to be talking about brook trout and their connection to clean water, and more about how that connection is there. And at the very end, there's going to be a Q&A session. I know we started a little bit late, so sorry about that. But we can have a few minutes for some questions at the very end. So please feel free to think of some questions, and then we'll discuss that at the very end. All right, so part one, brook trout biology and habitat. So this is the majestic brook trout. Scientific name, Salvelinus fontinalis. I believe I said that right. They are a freshwater fish species, and they're cold water obligates, so they require cold water to survive. And they're actually found in the more smaller first order headwater streams, and they can grow up to be 14 inches long, but usually in the wild they're more like 4 to 10 inches, 5 to 10 inches. And they can grow up to, they can be up to 8 years old, but in the wild it's more like 4 to 5. Their diet consists of mostly insects, and they're a very strikingly beautiful fish. They have a very unique color pattern. So they have a dark brown or green marbled pattern at the top with a very striking red belly, and their fins tips are black and white. And the special thing about them, oh, my laser pointer doesn't work on the screen, but they have these special spots, and they're actually light spots on a dark background. That's how you can sort of ID them. And they're red circles with blue halos, so they're very beautiful. And they're also native to the eastern United States. So this map here shows the distribution of brook trout in the continental United States. You can see that the orange coloration shows their native range from all the way from Maine down to the lower Appalachian Mountain Range and Smoky Mountains and everywhere in between, as well as some population in Michigan, in that area around the Great Lakes. And on the left side, we see with the dark red coloration, the non-native brook trout population. So this is where brook trout have been introduced to these other places, and they're not native to there. So these guys have been around since the last ice age, so they're native to this area. So they're not only a beautiful fish, they're also pretty important. They serve a recreational, economic, and cultural role. So they're Maryland's only native trout species. There are two other trout species I'm going to speak about in a bit, but this is Maryland's only native trout species. So that's one reason why they're important. Another is that they're very popular for fishing among anglers. So people who fish with a line and rod, it's very popular for that kind of fishing. And about 22,000 anglers fish for brook trout a year. And almost $10 million are spent on trip expenditures alone. So a lot of money is being, funneled into the recreational side of things. There are also many no-kill rules posted around different streams in Maryland. For example, we have this sign over here that tells anglers to don't kill brook trout. If you catch them, release them. And it's important, in order to do that, to actually ID the trout. So these are the other two trout species that are found in Maryland. So the top is the brook trout. You can see the light spots on the dark background and the red belly. And then we have the brown trout in the middle, which have dark spots on a light background. And then also the rainbow trout. So both of those two, brown trout and rainbow trout, were introduced from Europe to the Americas. So they're also the state fish of 9 states. So does anyone want to shout out a random guess as to what states these might be?
New Hampshire.
Yes, that's one. Any other guess? Yes, that's also one. Great.
Pennsylvania.
Yes. Oh, you guys are doing great. Any more?
West Virginia.
Uh-huh. West Virginia.
Michigan.
Yep, Michigan. Any other guesses? Yes, North Carolina too. Wow, like all right answers. Oh.
That's the only reason I would have said that.
I'm not going to talk about the quiz yet, but
What about Vermont?
yes, Vermont's one too. Any other guesses? Maine, yeah. So these are all the different, it looks like Maine is actually not one of the states, but so we have Pennsylvania, West Virginia, Michigan, New York, North Carolina, Vermont, Virginia, New Hampshire, and New Jersey. One that's not on here that I think should be on here is Maryland. But does anyone know Maryland's state fish? Yeah, no. I heard rockfish. That's the correct answer. Rockfish or striped bass are Maryland's state fish. So brook trout are very picky about the water that they like to live in. That's kind of an understatement. So some of the water quality requirements that they have is that they require cold water to live in. So the temperature of the water has to be less than 20 degrees Celsius, which is around 68 degrees Fahrenheit. Anything above 20 degrees Celsius stresses them out, and anything above 25 degrees Celsius kills them. So they're very sensitive to water temperature. Another thing that they're sensitive to is they require high dissolved oxygen. So just like how we require oxygen in the air to breathe, brook trout require a lot of oxygen in the water to breathe. Another thing is they require low turbidity. So turbidity is how cloudy or murky water is, and these guys require very clean, pristine water. So it can't be very muddy. It does stress them out. And as well as a pH between 5.5 and 8. So pH is a measure of how acidic or basic something is. 7 is neutral, which is about the pH of pure water. So these guys have a very limited pH range that they can survive in. So all this considered, they can actually serve as important bioindicators of cold, clean water. Because they are so sensitive to water temperature and all those water quality requirements, if we have them in a stream, we can have a pretty good guess that this stream has good water quality and it's cold and clean. Another thing that they're called is aquatic canary in the coal mines. So that term refers to back in the day, miners would take a caged canary or like a small bird with them into the mines. And if there's any leak of like carbon monoxide or high volumes of a toxic air or gases or anything in the mines, it kills the canary before it kills the humans. And that gives a sign for the humans to get out of the mine because it's dangerous in the mine. So in that sense, we can say that brook trout are aquatic canary in the coal mine. So if they're not found in a stream or a place where they should be, it's a bad sign of water quality. And seeing that there has been a significant decline in the brook trout population. So the graph on the left shows the predicted historic range of brook trout in Maryland. So you can see they're pretty much in all of those streams in the northwestern Maryland. And then the graph on the right shows the current range. So unfortunately, only a few little streams and pockets actually remain where brook trout are found naturally. So it's a pretty steep decline. So some of the threats that they face are urbanization. So as human development increases, people are building buildings and farms and creating these roads and impervious surfaces where stormwater runoff is exacerbated, as well as deforestation, so cutting down trees. So especially along those trees along rivers, riverbanks, those are called riparian buffers that really stabilizes the stream banks and prevents them from being eroded. But when those trees are gone, it creates a big erosion problem and increases the sediment and the dirt. It muddies up the water basically. Runoff, stormwater runoff, and also like agricultural pesticides and things like that, runoff from that. And pollution, so in particular acid mine drainage is a big problem. So this picture on the top right here of that orange muddy is actually pollution from mines. When heavy metals come into the surface and they mix with the water, it creates this really toxic substance for all the living things in the area. So if the mine waste is not properly treated, it does create a big problem. And then as well as competition with exotic trout species. So we talked about the brown trout and the rainbow trout that were introduced to the United States. So they're actually bigger than brook trout and they're more tolerant of different water quality ranges. They have a more varied diet compared to brook trout. So considering all of that, they often run out the native brook trout, so they outcompete them. As well as the big thing, which is climate change. So as global temperatures increase, the stream temperatures also increase, and brook trout have less and less places to go. And remember that they only can survive in water that's less than 20 degrees Celsius. This is going to become a pressing problem as years go on. So this map here shows the current distribution of brook trout in Maryland. So the green and the blue represent Maryland trout watersheds, and the red targets represent priority brook trout restoration watersheds. So you can see that there's three main ones in Maryland. So on the left is in Garrett County, and then we have one in the middle, which is northern Frederick County, and then over here is the Gunpowder watershed. And in particular, I want to bring your attention to the very top there, which we're going to talk about next. The Upper Gunpowder Falls watershed. So it's considered one of the last brook trout strongholds in Maryland. So this map over here shows the Gunpowder River. So the Gunpowder River is actually formed by the formation of two rivers going through Baltimore County. So you can see here there's Baltimore, 695, 83, the highways, and then the outlines of Baltimore County. This over here, the Gunpowder Falls River is actually around I believe 53 to 57 miles long, and it crosses over through the reservoirs and down to the northwestern Chesapeake Bay. And it's also joined by the small Gunpowder Falls River, so Gunpowder River. So these cross most of Baltimore County, and the upper Gunpowder Falls watershed is actually located at the very top. So this map shows that, again, at the very top of Baltimore County, kind of going into our Harford County, I believe it's called. So that shows you some of the map of the area there. So like I mentioned, there are only 3 significant brook trout populations remaining in Maryland, the Gunpowder being one of them. It's the second largest. It hosts the second largest brook trout population in all of Maryland, about 25% of the total Maryland population. So it's a pretty important area in terms of brook trout conservation. So this map shows a deeper dive into the actual distribution of brook trout. So you can see the green here shows where brook trout are currently present, and the red shows where there's no presence or it's unknown. So for not all the streams, but a lot of the streams do have brook trout present in them currently. And there's just some areas where they were historically present but have been wiped out. But as we can see, compared to other parts of Maryland, this is doing a pretty good job in supporting the local brook trout.
So is that just a one-time shot analysis of now?
I believe this is from 2016. Yeah, this map is from 2016.
Okay.
So what is it about the Gunpowder that makes it such a good habitat for brook trout? Well, one is that it's less urbanized. There's less people living here. There's less development, farms and things like that, meaning there's less pollution and human activity in the area. Another thing is that there's more tree cover. So these trees are present on, like I was mentioning, those riparian buffers, which are trees that are found on the banks of the streams that are really stabilizing the stream banks. Those are still mostly intact, you can say. They have not been cut down. There's also less competition with brown trout and those other trout species. So there's less competition. Brook trout, they sort of have a stronghold over there. Okay, now I'm going to switch gears a little and talk about brook trout and their connection to clean water. So you might be wondering what any of this has to do with clean water. Sure, there's a lot of species that are endangered or vulnerable. There's a lot of fish species, that are declining. But what is it about brook trout specifically that has a connection to clean water? So I want to bring your attention to the Pretty Boy Reservoir. So the upper Gunpowder Falls watershed that we talked about over here on the left actually is a major tributary to the Pretty Boy Reservoir. So a lot of these streams feed into the reservoir. And fun fact, the name Pretty Boy Reservoir, I was kind of curious where that came from. Actually, an early settler had a horse named Pretty Boy who drowned in a nearby creek. So they named the reservoir after it. It's not a person. I was curious where that came from. So the Pretty Boy Reservoir supplies a lot of our drinking water. So it's a major water source for the Baltimore metropolitan area. It's often called a drinking water source or a reservoir. And it supplies about 61% of the city's drinking water. So that's a considerable amount. The water that we're drinking, comes from the Pretty Boy Reservoir, which is fed by the upper Gunpowder Falls watershed. So the Baltimore area is home to about 500,000 people, and the Baltimore metropolitan area is home to about 2.8 million people, almost 3 million people. So that encompasses most of Baltimore County, Baltimore City, parts of northern Anne Arundel County, as well as eastern Howard County. So even though the reservoir itself is located in northern Baltimore County, the reservoir is actually owned by the city of Baltimore, and the surrounding land is also owned by Baltimore City. So this shows a picture that there's a direct connection between the reservoir, this place that's not as inhabited or urbanized, all the way down to the Inner Harbor. So there's a connection there. So the fact that there are brook trout found in the upper Gunpowder Falls watershed is a pretty good sign of water quality. You know, these guys are sensitive. They're bioindicators of cold, clean water, and that the fact that they're found in this watershed, it's a good sign of water quality. But will it last? This map over here shows the trend in where brook trout occupancy or distribution. So not distribution, occupancy. So the red shows that the populations have been greatly reduced. The yellow is reduced, and the green is present or intact. And as you can see, overwhelmingly, it's red. You know, these populations are decreasing. Their numbers have been going down in recent years. There's only a small population in Garrett County in the west here where the population is intact, but overwhelmingly it is declining, especially up here in the Gunpowder Falls watershed. The population of brook trout is declining. So it's really important that we preserve and expand existing brook trout populations. But to do this, it requires more intensive and efficient monitoring and restoration efforts. So some of the tools and technology that are actually used to see what areas need to be conserved is electrofishing and environmental DNA. So I'm going to be talking briefly about each of these things. So both these technologies can tell us where brook trout are, because we need to know where they are before we actually start creating management or restoration plans. So electrofishing is one of the more traditional methods. So this picture shows what a typical electrofishing survey would look like. So this occurred in the Gunpowder in 2015. So what happens is that a weak electric current or electric field is created in a section of water, and it temporarily stuns the fish and kind of brings them to the top. They're attracted to the anode, and it brings it to the top. And people scoop them up with the nets, and they measure them, they count them, they ID the fish, and that gives a pretty clear answer to how many brook trout are present in a section of stream. And these electrofishing surveys are usually conducted by the Maryland Department of Natural Resources or the Maryland Biological Stream Survey. The thing about electrofishing, though, is that it can't be all intensive. It's really impossible to go out to every single stream and conduct these surveys. They are costly, they require a lot of people, and it's very labor intensive. So it can't be practical, especially for these small streams. Many of these small streams are left unsurveyed, or any streams that are remote and hard to access with all this equipment. You know, these streams and rivers might be prime brook trout habitat. So it's important to not ignore the smaller streams, because you never know where brook trout can be found. So there has to be a better way of doing this, and we can use deoxyribose nucleic acid, or DNA, which is the blueprint of all living things. And the thing about DNA is that organisms are constantly shedding it into the environment through hair or skin, fur, feathers, feces, eggs, all these different sources. And it kind of leaves a trace in the environment of where an organism has been. And scientists can do detective work and actually isolate these DNA that's found in the environment and figure out where a species has been. So that is what environmental DNA is. So the E stands for environment. As the name suggests, it's just DNA left over from an organism that is found in the environment. So this is a relatively non-invasive and cost-effective new technology. So there's no need to actually see the organism or capture them or observe them. You can just take a sample from the environment and get an idea of what species has been there. So how this really works is that scientists go out to the field and take a sample from the environment. This can be a water sample or a soil sample, ice core, even the air. And then we can go back to the lab and isolate and extract these DNA strands that are present in the sample. And this can be sort of a barcode that matches with a specific species. And we can identify a range of organisms, from the largest land animals, mammals, to the smallest microscopic bacteria. So this just shows, fish, plants, bugs, or single-celled organisms, prokaryotes. It has a very large and wide applicability. But there are a lot of unknowns when using this technology. So this technology has only really taken off in the past 10, 20 years, I would say, so it's relatively new. And this is the research that I'm focusing on for my master's thesis. So I'm trying to improve eDNA methods specifically for brook trout conservation, so seeing how different factors like temperature and distance affect how well we can detect brook trout eDNA. So this has been the research that I've been working on the past year. I've been going out to different sites. I've been doing lab work and also stream lab work. So this has made me really passionate about the use of eDNA, and it's a pretty exciting new field. So a bunch of other organizations have also been focused on brook trout conservation, up from the national and federal level down to local efforts. So this just gives some examples of that. So at the national level, we have the USGS, the U.S. Geological Society, who I'm also collaborating with for my research, National Park Service, Chesapeake Bay Program, among others. State efforts include the Maryland Department of Natural Resources, who do a lot of these electrofishing surveys, among other things, as well as local efforts like Trout Unlimited, and the Gunpowder is also doing some work there, the Gunpowder Riverkeeper. So there have been many management efforts for protection and restoration of brook trout, including fishing regulations, so things like these signs being posted at different streams, telling anglers to not kill the brook trout, essentially, and pollution remediation, especially for those acid mine drainage, so preventing that toxic sludge from getting out into the streams. Habitat enhancement in the form of reinforcing the banks of streams as well as planting trees, and water quality improvement projects, and even outreach to decision makers and stakeholders. So to actually make a bold effort in protecting brook trout, we have to be very assertive about towards our government entities and lawmakers who are actually, have the authority to protect different areas where brook trout are found. But you guys might be wondering, not everyone can be directly involved with this kind of work, but there are things that any person can do. So that's where there's some volunteer opportunities through the Gunpowder Riverkeeper. So it's just some pictures that I got from the Instagram page of the Gunpowder Riverkeeper. Landscaping, so planting trees and things like that. Trail maintenance, so cleaning up trails, making sure pollution doesn't get into the streams. Tabling, advocacy work, staffing, and cleanups. So there's a bunch of opportunities that anyone can spend a day doing, or they can commit to it long term, but everyone can do something small. Or even coming out to these educational opportunities like this talk here today, just raising awareness for this issue is a big step in the right direction. So I hope you guys understand that we should be protecting our waterways, not just for the brook trout, but also for our drinking water supply. From forest to faucet, the brook trout and us, we all rely on the same water, so it's our duty to protect it. Thank you for listening. So I'll take any questions. Have a couple minutes for questions. Yeah.
So in those watersheds where you showed that the brook trout had been introduced,
Mm-hmm.
what's their fate like there? Are they similarly threatened in reducing in numbers, or are they doing better there?
So the brook trout that have been introduced in Western Maryland, for example, they're actually doing pretty great for themselves. And in fact, they're actually trying to get rid of them in many of these streams. So they've often taken over niches that have been left behind by other native fish. So it's kind of funny how over here in the eastern United States, we're really focused on conservation efforts and trying to save them. But over there, they're doing great. And in fact, people don't want them over there. So
Send them to us.
Is it because they're taking over the native population that they don't want them there, or I'm confused?
I believe, I do not know exactly why, but I can look into that. Good question. But I imagine the reason why a lot of people would want them gone is because they're causing ecological destruction in some way or another, and that might be because they're displacing the native fish.
Too many numbers.
Yeah.
So, question about that, numbers. The environmental DNA, does that give you a quantifiable amount?
Yeah, it does. So yeah, we can use a method called quantitative polymerase chain reaction, qPCR, kind of a mouthful, but this actually allows us to amplify and actually quantify how many, estimate how many strands of DNA are present in a sample, and we can sort of extrapolate that into seeing how much, perhaps how much fish are occupying that, where the sample came from in a stream.
And is that cumulative, or is it time parameter defined? Does the eDNA study, does that give you quantifiable time as well?
That does not actually. That's a great question too. So, so you're asking if we can actually tell when that DNA
Right.
came into the environment.
Or is it cumulative? Is it a
Mm-hmm.
time period? Or?
Yeah, it's actually not.
Okay.
You can't really tell when that DNA came from the organism. It's more of the the DNA that's present in the environment. We just collect that sample, and there's no way of actually telling when was this DNA deposited in the environment, or we can have some evidence of the more degraded some DNA strands are and harder to amplify. Then we can kind of tell maybe it's been there for a while. It's been breaking down in the environment from sunlight or things like that, from environmental factors. But as of now, we can't really, we don't really have a time frame of when that DNA came out. Yeah.
Is there a chance with the eDNA that you could like miss DNA and like not detect the fish even if they're there just because of where you sampled?
Yeah, for sure. So false negatives are a big issue in the eDNA world. So there might be an issue of that we're just not sampling in the right spot. If we're going out and taking like a water sample, we might not be in the right spot, or maybe we're not sampling, we're sampling too close to the banks and we have to be in the center of the stream, all these factors. It really does make that hard in the sample collection process. So it's definitely a possibility that we can miss DNA, yeah.
Are there false positives too?
Yeah, and that's more of a problem with primer design.
Yeah.
So when we're doing qPCR, we need a way to actually find where the brook trout DNA is and sort of sift through all the other DNA and just find the brook trout DNA. And if there's a problem with that process, we can get false positives that there is DNA here, but So for example, taking a soil sample and we find shark DNA, that's an example of a false positive. Yeah, that can totally happen. Yeah.
So when you're out in the field taking these samples, like what volume of water are you collecting?
Mm-hmm. That's a great question too. There's no one silver bullet for the perfect amount of water, but what we've recently been doing is collecting at least 2 liters of water. The U.S. Fish and Wildlife Service actually has a standard for their eDNA collection methods where they try to get a minimum of 1 liter or 2 liters. I have collected 3 liters of water in the past. I've also collected 300 mils, so way less than 1 liter. I have gotten DNA from some of those samples too, but generally the more water you do sample, the better chance of getting the DNA that you want to see. Mm-hmm. Does that answer your question?
It does.
Mm-hmm. Yeah.
Have you ever gone snorkeling?
No. No.
Highly recommend.
Do you want to?
I do, but I'm not a great swimmer.
Like shallow rivers.
Right,
Actually
just
hold
thinking
on
off
to
the
a rock.
street.
Okay. Yeah, I'm down.
You can just like crawl up.
Yeah.
With things like turbidity being a big issue for brook trout, I don't know a lot about turbidity. Is that something that like humans can easily change? Like I'm just imagining like if you go in the river,
Mhm.
that might like be a problem for them.
Yeah, well, turbidity is a measure of how much light can pass through water. So if there's more things or stuff like sediment in the water, it's harder for the light to pass through. So essentially, that is can be caused by erosion or high sediment load, often from human activity. I'm not exactly sure if there's a way to reverse that, but that's a great question. I guess planting those trees in riparian buffers prevents the soil erosion from occurring, and over time, making the turbidity not as bad, so clearing up the water a bit. So I do know in the Chesapeake Bay that oysters are filter feeders, and they filter out the sediment and all the gunk from the water, clears it up. Mussels do that too, but I'm not sure about these smaller streams here. That's a great question. Yeah.
You said you noticed you were in, which river were you in was it the Shenandoah River or the water,
Yes, yeah.
And what did you notice about the turbidity there?
So I was out in the Shenandoah National Park last week on Friday collecting eDNA samples for brook trout, and Shenandoah is a great spot. There's a lot of brook trout there. There's a saying that if you throw a stone over your shoulder, you'll land in a brook trout filled stream. So brook trout are pretty prevalent. And I did notice something while I was out there, that the water is very clear, very pristine. We were trying to measure turbidity using one of those turbidity tubes with a Secchi disc, which is a way to measure the clarity of the water. And it was just too clear to have like a clear, no pun intended, clear idea of the turbidity. So it was hard to quantify that just by visualizing. So that's when I made the connection that, this clear water makes perfect sense why brook trout are so abundant here.
And then it's in a national park, right?
Where it's protected.
Where it's protected.
Exactly. A lot of trees, very protected. Mm-hmm. Yeah.
What's the biggest limiting factor to sort of processing the eDNA data? Is it getting enough samples? Is it running it through whatever laser machine you're putting on?
Yeah. Biggest setback to collecting eDNA
Or
samples.
just like what would help you like speed it up or do more?
There's a lot of things that can go wrong when collecting eDNA. So there's different precautions we can take in the field and in the lab to maximize our chances of getting a higher hits of DNA. So when we're out collecting, it's collecting in the right spot. So if we have an idea of this, for example, brook trout, if we have an idea of brook trout are found in these streams, there's a good chance that we'll be able to get the brook trout eDNA. So the number of the organisms or individuals present in the area, the more fish there are, the more DNA there'll be. So that's something, as well as collecting higher sample volumes of water. So maximizing the amount of water that we're actually filtering through will give us a better chance of actually collecting more DNA to work with. And then making sure that DNA is properly stored and not, degrading while we haven't filtered it or extracted it yet. And then in the lab, there's also things we can do like using good primers. So primers, like I mentioned, is how we can actually single out the brook trout strands of DNA from everything else. So if we have a more specific, optimized primer that's better at finding those strands of brook trout eDNA, it can give us a better chance of getting reads from the samples that we have. So those are just some of the ways. As well as inhibition. Inhibitors are a big problem when we're taking water samples. So there's stuff in the water that kind of gets in the way of the primers doing their job. So that's something we need to be looking out for. So there's a lot of things we can do along the way to better our chances. And again, this is active research. This is the research I'm doing right now. So there's a lot of different factors we can be looking at, but we're getting there. Yeah. Great. Yeah.
So what is the main reason why the fishermen use it most for the fishing?
Why do people love fishing brook trout?
Why they use it for fishing?
Use it for fishing?
Yeah, to catch the other fish.
So brook trout aren't used as bait, but a good bait for catching brook trout, if you want to go out and catch brook trout, is they're insectivores, so they eat a lot of bugs and insects and stuff. So, and they eat lots of different kinds of bugs. So if you do want to maximize your chances of catching a brook trout, you can use different types of insects, see what insects are present in that specific area where you're fishing. That's one way you can catch brook trout. But I've not heard of brook trout being used as bait for other. I don't fish, so I don't really know. So.
Because it's so tiny.
Mm-hmm. Yeah, I mean they are 4 to 10 inches, so they're not.
I think there was a picture up there where they looked very small. Those must have been like young.
Yeah, so baby brook trout. Yeah, exactly. Like the yearlings are quite small. I have seen these in the wild where pretty small. You know, they're in a group, but the adults can get bigger than that. Mm. And especially if you go and get hatchery fish, so fish that have been hatched in a laboratory or controlled setting, those can get a lot bigger than the wild trout. Yeah.
In your experience, are hatchery-raised brook trout as beautiful as wild brook trout?
No. I would say the brook trout that are found in the wild are a lot more beautiful. Their colors are a lot more stunning. I saw some at Shenandoah last week. The red underbellies are so striking in the water. They're beautiful fish. But that's not to say that the hatchery fish aren't beautiful too, but their coloration is a lot duller, I would say. Yeah.
Okay, going off of that, if I would imagine in a controlled setting, they're like perfectly, cared for, and that's why they're bigger. Do we have an idea of why they might be more colorful in the wild as opposed to the controlled setting?
Well, one idea I have is that their diet is more varied in the wild. So usually when they're in the tanks, the hatchery fish, they're just fed pellets. Just throw some pellets in there and they eat them up. That's all they eat, 2 times a day. So that's one of the main reasons why their coloration might be a lot more striking in the wild.
And the water that they're in.
Mm-hmm. Yeah.
It's more clean.
Perhaps, yeah. Great. If there's no more questions, I have my email up here, aimanraza@umbc.edu. If you want to connect later on or ask any more questions, feel free to reach out. And before we end off, if you would like, you can take the quiz again, and hopefully by now you should fly through the quiz, get 100%, although I'm not grading you guys. But all right, thank you, everyone.
Thank you.